Sodium-dependent glucose cotransporters are a family of glucose transporter found in the intestinal mucosa of the small intestine and the proximal tubule of the nephron. They contribute to renal glucose reabsorption. In the kidneys, 100% of the filtered glucose in the glomerulus has to be reabsorbed along the nephron. In case of too high plasma glucose concentration, glucose is excreted in urine. Because SGLT are saturated with the filtered monosaccharide, glucose is never secreted by the nephron. Types: the two most well-known members of SGLT family are SGLT1 and SGLT2, which are members of the SLC5A gene family. In addition to SGLT1 and SGLT2, there are 5 other members in the human protein family SLC5A, several of which may also be sodium glucose transporters. SGLT2 inhibitors for diabetes. Inhibition of SGLT2 leads to a reduction in blood glucose levels. Therefore, SGLT2 inhibitors have potential use in the treatment of type 2 diabetes. Several drug candidates have been developed or are currently undergoing clinical trials, including dapagliflozin, approval rejected in 2012 by Food and Drug Administration due to safety concerns. However, after resubmitting, additional clinical data is under review with December 12, 2013 as PDUFA date, but marketed in Europe and Australia. Dapagliflozin was the first SGLT2 approved anywhere in the world in 2011 by the EU. Canagliflozin, approved in the United States and Canada. Ipragliflozin, in Phase 3 clinical trials. Tifogliflozin, in Phase 3 clinical trials. Empagliflozin, approved in the United States. Sergliflozin etabinate, discontinued after Phase 2 trials. Remogliflozin etabinate, in Phase IB trials. Function: firstly, the Na+/K+H base pump on the basolateral membrane of the proximal tubule cell uses ATP to move 3 sodium ions outward into the blood, while bringing in 2 potassium ions. This creates a downhill sodium ion gradient inside of the proximal tubule cell in comparison to both the blood and the tubule. The SGLT proteins use the energy from this downhill sodium ion gradient created by the AT base pump to transport glucose across the apical membrane against an uphill glucose gradient. Therefore, these co-transporters are an example of secondary active transport. Both SGLT1 and SGLT2 are known as symporters, since both sodium ions and glucose are transported in the same direction across the membrane. Discovery of sodium-glucose cotransport. In August 1960, in Prague, Robert K. Crane presented for the first time his discovery of the sodium-glucose cotransport as the mechanism for intestinal glucose absorption. Crane's discovery of cotransport was the first ever proposal of flux coupling in biology. See also, cotransport, cotransporter, glucose-galactose malabsorption, renal sodium reabsorption, discovery and development of SGLT2 inhibitors. References. External links: Sodium-glucose transport proteins at the U.S. National Library of Medicine. Medical subject headings.